Welcome to Episode 6 of Epigenetics Explained. Today we will learn how our ancestors' lifestyles and diets might have had an impact on our own epigenome. That is, how epigenetic inheritance works. Multi-generational inheritance is a type of inheritance that involves multiple generations being simultaneously exposed to environmental factors that cause phenotypic changes. This can take place in mothers or fathers if the cytoplasm of reproductive cells, or germ cells, are exposed, and in pregnant mothers only if the fetus itself and all the intrauterine conditions, which is the environment of an embryo, are exposed. Environmental exposures may cause changes in the levels of nutrients, hormones, and other environmental signals that the fetus is exposed to, resulting in phenotypic changes. These changes can last up to 2 generations if a male and his sperm are exposed, or 3 generations if a pregnant female is exposed, as environmental factors can affect the germ cells of the fetus. However, multi-generational inheritance does not involve changes to the epigenome. Transgenerational inheritance involves the exposure of just one generation to certain environmental factors, the resulting epigenetic changes of which are inherited to offspring. Transgenerational inheritance has occurred if the epigenetic changes survive through 2 waves of epigenetic reprogramming. This is essentially the removal and replacement of methyl marks, and the first wave occurs a few days after conception, while the second wave occurs while the fetus is developing and resets most of its epigenetic changes. These changes must be present in more than 2 generations following a male's exposure or more than 3 generations following a female's exposure to be deemed evidence of transgenerational inheritance. Now, there is a notable experiment conducted by researchers from Washington State University which demonstrates transgenerational inheritance in mice. The chemicals shown here are called methoxychlor and vinclozolin, and they are endocrine disruptors and interfere with the development of the reproductive system. Researchers injected these chemicals into pregnant rats during the period in which the embryo's reproductive systems develop. As expected, the male offspring were born with abnormal testes that produced weak sperm, of which there were too few. However, one of the researchers accidentally bred these pups with normal female rats. As a result, more than 90% of the grandchildren had testicular abnormalities. The researchers then decided to breed 4th and 5th generations, the males of which suffered abnormalities similar to those of their ancestors. Follow-up studies have found that the great-grandchildren of rats exposed to fungicides consistently had abnormal DNA methylation patterns in their reproductive cells and organs. This suggests that the initial environmental disturbance exerted its effect by interfering with normal DNA methylation, and that transgenerational inheritance had taken place. I hope you've enjoyed this episode on epigenetic inheritance. It's a little different from previous episodes, but to be honest, this video has been my favourite to produce so far. Stay tuned for episode 7, which will be about the epigenetics of cancer.